Welcome to Biotechnology World. Now our topic primer designing. Introduction to primer design. 1. Primer design is a crucial step in PCR, polymerase chain reaction and other molecular biology techniques involving the selection of short DNA sequences that flank the target region of interest. 2. Primers serve as starting point for DNA amplification and are essential for the specificity and efficiency of PCR reactions. Objective of primer design. 1. The primary objective is to design primers that specifically anneal to the target DNA sequence, minimizing non-specific amplification and ensuring high specificity. 2. Primer design aims to optimize various parameters such as primer length, GC content, melting temperature, and avoidance of secondary structures. Major steps involved in primer designing. 1. Target sequence identification. Identification of the target DNA sequence or gene region to be amplified, typically through sequence alignment or database search. 2. Primer design software. Utilization of primer design software or online tools that employ algorithms to generate primer meeting specified criteria. 3. Primer parameters optimization. Optimization of primer parameters such as length, usually 18 to 25 nucleotides, GC content, typically 40 to 60%, and melting temperature to ensure primer specificity and efficiency. 4. Avoidance of secondary structures. Here, minimization of primer secondary structures and primer dimer formation, which can interfere with PCR amplification. Primer design guidelines. 1. Specificity. Primer should be specific to the target sequence, avoiding regions of homology with non-target sequences to prevent non-specific amplification. 2. TM matching. The melting temperatures of forward and reverse primers should be closely matched to promote annealing efficiency during PCR. 3. GC content. The GC content of primer should be optimized to ensure stable primer binding and efficient amplification. 4. Primer length. Primer should typically be 18 to 25 nucleotides in length to balance specificity and efficiency. 5. Avoidance of self-complementarity. Primer should not have significant self-complementarity to prevent hairpin formation and primer-dimer artifacts. Primer designing applications. 1. PCR amplification. Primer design is essential for PCR-based amplification such as gene expression analysis, genotyping, mutation detection, and cloning. 2. Next generation sequencing, NGS. Primers are also used in NGS library preparation protocols for targeted sequencing and amplicon sequencing. 3. Site-directed mutagenesis. Designing primers for introducing specific mutations or modifications into DNA sequences for functional studies or protein engineering. Challenges and limitations. 1. Sequence variability. Variability in target sequences such as polymorphisms or sequence repeats can pose challenges in primer design and specificity. 2. Secondary structures. Complex secondary structures within the target sequence can hinder primer annealing and PCR amplification. 3. Non-specific amplification. Non-specific amplification due to primer cross-reactivity or template contamination can lead to false positive results. Future directions. 1. Advanced primer design algorithms. Development of advanced algorithms and software tools for primer design that consider additional parameters and optimize primer performance. 2. Multiplex PCR. Advancements in multiplex PCR primer design for simultaneous amplification of multiple targets in a single reaction, enabling high throughput applications. 3. Customized primer libraries. Creation of customized primer libraries tailored to specific experimental needs and target sequences for improved efficiency and reproducibility. Thank you.